Okay, so let's start the class. Hi, good evening everyone. So today is the fourth problem solving session of the course Experimental Biotechnology. So till now we have discussed about different techniques about electrophoresis. So today we will be discussing about chromatography. We will start discussing about chromatography. So whatever doubts you have, you can ask me, and along with that I'll be solving some questions which are related to your assignment, so that will help you to solve the assignment questions as well. And feel free to stop me wherever you want to ask any question. Okay, you can either write it in the chat box or you can ask me directly, that's your wish. Shall we start the class? I hope I'm audible and everyone can see my screen as well. If one of you, yeah, thank you. So we'll start now. So I'll start with the first question. Extraction of butanol from fermentation broth, that is water based, which has butanol concentration of 20 grams per liter, is performed by adding dichloromethane and mixing it, and after mixing and phase separation, the organic fraction is estimated to have butanol concentration of 5 grams per liter. So what is the distribution coefficient of butanol? So we have four options here: 100, 4, 0.25 and 25. So we have we want to extract butanol. The extraction process is done by using DCM, which is an organic solvent. So we mix it with DCM and let the organic and aqueous layers separate, and some butanol goes into the organic layer. The amount of butanol in our broth was 20 grams per liter, and the amount of butanol, the concentration of butanol in the organic fraction after the phase separation is 5 grams per liter. So what will be the distribution coefficient of butanol? If anyone knows the answer, you can answer it in the chat box. Anyone wants to answer? Nobody? So we have to find the distribution coefficient. So let's understand what distribution coefficient is first. So distribution coefficient basically is how much one component that you are trying to separate, so that one component, how much it is being separated in two different phases. Now in this case we have an organic phase and an aqueous phase, right? So how much butanol is going into organic phase versus how much butanol is going into aqueous phase, that the ratio is basically distribution coefficient. In very simple words, how much that one component that you are trying to extract or that you are trying to separate from the mixture, how much it is getting separated between two different phases. Okay, so this is in very layman language. So now let's see. So we have 20 grams per liter. The concentration of butanol is 20 grams per liter. So out of that 20 grams per liter, 5 grams per liter is going into organic phase. That means from the total, 5 grams is going into organic phase. So I have to take the ratio of how much butanol is in the aqueous phase, how much butanol is in the organic phase. So simply we have to divide the concentration of butanol in phase 1 divided by the concentration of butanol in phase 2. That is the simple way to calculate the distribution coefficient. So simply we will have 20 grams per liter divided by 5 grams per liter. So that will give us the answer 4, that is option number B. Okay, we had some answers. People said option C. How did you land in option C? Did you calculate the percentage instead of the fraction? So basically, distribution coefficient is basically how much the molecule is distributing between two phases. So simply you have to divide the concentration in phase A divided by the concentration in phase B. Now we knew that in phase A, that was the aqueous phase, we had 20 grams per liter, right? The aqueous phase had 20 grams per liter and the organic phase had 5 grams per liter. So simply the ratio will be 4. So the distribution coefficient is 4. Okay, you did organic by aqueous. So in that case, when you have been asked like what is the distribution coefficient of butanol, you have to understand we can take it in, we can consider any phase as phase A and any other phase as phase B. But generally when you have given a question like this, so basically what you are trying to do, you are trying to extract butanol from the aqueous phase using an organic solvent, right? So your aqueous phase, that is the starting material, will automatically becomes phase A because butanol was initially in the aqueous phase. So when we are doing chromatography, generally we take stationary phase as the phase A and mobile phase as the phase B, because stationary phase is there, our analyte binds to the stationary phase and then we pour the mobile phase with which slowly we elute our analyte. So similarly, when we are doing this, mostly we take the first phase that is in contact with our component as the phase A and the second phase that gets in contact with the component as phase B. Okay, so in this case we had butanol in aqueous phase, right? So we will take aqueous as the phase A and we will take organic as the phase B. If in the question it was specially mentioned like what is the distribution coefficient of butanol in the organic phase, then we will have to take the reverse of it. Okay, so you were not wrong, like you can take distribution coefficient is just a ratio, so you can consider, it's up to you, you can consider phase A as organic or aqueous, but generally the notion goes like wherever your analyte is present first, we take it as phase A and then second phase is the one that comes later in the system. Okay, so that's why here also in column chromatography what do? We have a column. So we have something like this, a column in which some form of resin or bead is packed in it. Okay, and we pour our analyte, whatever we want to separate. Let's see, we have a mixture of A and B. I want to separate the mixture of A and B. So I will pour the A and B mixture onto the column. A and B will bind to the column with separate affinity based on whatever technique we are doing and whatever the attachment process is there, like principle is there. So they will attach to the column based on that. So the analytes, the A and B are coming in contact with stationary phase first. So we will take phase A as the stationary phase and then we will pour our mobile phase buffer into the column to separate A and B. So let's say B elutes first and then A elutes later. So the mobile phase is coming in contact later. So mobile phase we generally consider it as phase B. Okay. Okay, so if that is clear, let's move on to the next question. The partition coefficient Kd Option A depends only on the physical properties of the molecule. Option B depends only on chemical properties of the molecule. Option C depends on both physical and chemical properties of the analyte. And option D, it is independent of the physical or the chemical properties of the analyte. So now you have understood what partition coefficient or a distribution coefficient is. So tell me if it is dependent on physical properties, chemical properties, both or none. Okay, Suman has answered option C, that it depends on both physical and chemical properties. Okay. Okay, so all of you are agreeing with option C. So that is the correct answer. It depends on both physical and chemical properties of the analyte. So what all physical and chemical properties are there? Let's see. So we have different physical properties that can be utilized to separate two molecules. So first is molecular weight. So let's say we have mixture of two components A and B. I have a mixture in which component A and component B is there. I want to separate two of them and they have a very different molecular weight. Okay, let's say A is 90 grams and B is 9 grams. Okay, so you can see there is a huge difference in their molecular weight. Okay, so that means A will be a bigger particle, the molecular size will be bigger, and B will be a smaller particle. So we can easily separate them out using some kind of filtration technique where the smaller particle can pass through and bigger particle will be stuck, or the bigger particle will elute out from the column and the smaller particle will be eluted from the column later. So something like that we can employ a chromatography technique or a separation technique. So whatever medium we are using, if we are using a gel filtration chromatography or whatever medium you are using, if your A is interacting more with the with that medium, that means the partition coefficient for A is higher than partition coefficient of B. Okay, so similarly, like basically partition coefficient will be based on molecular weight if the separation is based on, if we are doing the separation based on the size. Similarly for boiling point, now if I have a mixture of A and B, the boiling point of A is very high and boiling point of B is very low. So what I can do, I can take them in a solution in a beaker where I have A and B and I'll start heating it. Okay, I'll start heating it. What will happen? Since B has a lower boiling point, B will evaporate. So I can collect a B using a condenser. So let's say this is a condenser. So I have the container where we have A and B. So since B has a lower boiling point, B will boil first and I can collect out B, and then in the container I'll be left with only A. So this is how I can, in another container I can collect the analyte B. So this is how, using the boiling point as a criteria, I can actually separate two molecules in a mixture. So that means there also partition coefficient is depending, because the analytes A and B are partitioning between two phases. Here what are the two phases? Liquid and gaseous phase. So we have two phases, right? Similarly, freezing point will work the same way boiling point is working. If you freeze a compound, if both of them have different freezing points, one will freeze first, second will freeze later, so you can separate them out. Crystallization will also work in the same way. If both of them have different crystallization properties, one will crystallize faster than the other, and once, like let's say A and B components are there, A crystallizes first, you can take out the crystals, filter out the crystals, and you will have B in the solution. Okay, so crystallization can be used. Solubility, you saw the example of butanol already. We had butanol in the organic phase, aqueous phase. If I know that butanol is more soluble in some organic solvent, I can simply add the organic solvent. Butanol will go into the organic phase because it is more soluble in organic solvent than in the aqueous phase, and I can separate the aqueous and organic layer, and that's how I can extract or separate two analytes based on their solubility. Then density, again same concept, based on if one is more dense than the second analyte, I can take out one analyte from the other based on density. Now coming to the chemical properties. So all these properties are physical properties. So what do you mean by physical and chemical properties? I hope you understand the difference between physical and chemical properties. So physical properties are basically the properties that are not changing with the changing the chemical form of the compound. So there is no chemical change happening. So that is those properties that do not change the chemical form of a compound. So after boiling a compound, you are not changing the chemical formula of that compound, right? If you boil water, it will still be H2O. It will be in steam form, but it will still be H2O. So, or if you freeze water, the ice will still be H2O. It will be in a crystalline form, but it will be H2O. So that's how that's what we mean by physical properties. But by chemical properties, we mean the chemical nature of the compound is different. So the chemical formula of that compound will be different. So there can be different functional groups. So if you have phenol will be having OH. If you have let's say toluene, so it will have another functional group. It will have CH3. If you have nitrobenzene, so it will have a nitro group. So similarly, different chemicals will have different functional groups. So that is the difference between their chemical property. Based on differences in chemical properties also, you can isolate two different compounds. Just a second. Yeah. Based on two different based on the chemical properties, you can actually separate two different compounds in a mixture. Now the second chemical property that is there is reactivity towards other reagents to form complexes. So now let's say I have a column that has special property towards a particular chemical group or towards a particular component. So if I have a mixture where that particular component is there and other things are there, when I pass it through that column, only that particular component that has a reactivity towards the functional group will react to it, and other things will get out of the column, and then I can separate my analyte. So something like that, I can use the reactivity towards other reagents. I can make a complex of that component, and then I can again regain the component of interest by breaking that complex. Okay, so these are chemical reactions happening. So that's why these are chemical properties that are affecting the partition coefficient. I hope that is clear. Moving to the next question. Given below are the partition coefficients of various compounds. Compound A, Kd is 5. Compound B, the Kd is equal to 2. Compound C, Kd is equal to 10. And compound D, Kd is equal to 7. Mixture of these compounds is passed through a chromatography column. What will be the order of elution of the individual compounds? So you have different partition coefficients for each compounds, and now if I pass all the mixture of these compounds through a chromatography column, what do you think? Which will elute first, which will elute last? What would be the order of elution? Okay, we are getting answers. We have option A as the answer. Anyone else wants to answer? Okay. Shahina says option D. Okay. Anyone else? Okay. So, here let me ask you one question first. So, till now we have discussed about partition coefficient. So, what do you think will like if I have a partition coefficient of 2, what do you think it means? It means two parts of like compound B is divided into two different phases, such that two parts of compound B are in the phase A and one part is in phase B. If you remember, I told you Kd is equal to concentration in phase A divided by concentration in phase B, right? So, if I have a compound whose partition coefficient is 2, that means two parts of it is in phase A and one part is in phase B. If I have a partition coefficient of 5, that means 5 parts of it is in phase A and one part is in B. Similarly, if something has partition coefficient as 10, that means 10 parts of it is in phase A and one part is in phase B. And for the last compound, 7 parts are in phase A and one part is in phase B. Now, if you remember, I told you we generally consider the stationary phase as the phase A and mobile phase as the phase B. So, if I have a column, and we have the stationary phase. So, now tell me, phase A is the stationary phase. So, for compound A, let's start with compound A. Compound A, 5 parts of compound A is in stationary phase. Okay. Now, for compound B, only 2 parts are in stationary phase. That means wanted to write B. Yeah. So, that means A is interacting more with the stationary phase than B, right? For A, 5 parts are in stationary phase, whereas at the same time B has only 2 parts. C has 10 parts, so C is interacting way more than compound A and B. Then you have D, which has 7 parts. That means D is interacting between C and A with the stationary phase. Now, I add the mobile phase. So, what do you think will elute first? If C is interacting most, then D, then A and then B, that means when we will take the elution, since B is interacting least with the stationary phase, B will get eluted first, right? So, B will get eluted first, then we will have elution of A, then we will have elution of D, and then we will have elution of C. So, do you understand what does a partition coefficient means? Whatever the number is there, that means those many parts of that particular compound are interacting with the stationary phase. So, if something has a higher partition coefficient, that means it is interacting more with the stationary phase. If something has a lower partition coefficient, that means it is interacting less with the stationary phase. So, that means the order of binding is affected by the partition coefficient. Okay. Now, compound C had a very high partition coefficient. That means the compound C can interact with the stationary phase the most. So, when mobile phase will be put, the compound C will, since it is interacting with the stationary phase most, it will keep on interacting with it and get eluted out of the column the last. And for compound B, since the partition coefficient is least, that means it does not want to interact with the stationary phase. Once I add the mobile phase, it happily comes out of the column because it does not want to interact with the stationary phase that much. So, do you understand? So Shahina, do you understand why the order will be BADC? Yeah, good. So the correct answer is option A. Anyone has any doubt in the concept of partition coefficient or any of these questions, or any other question if you have in mind? I can try to solve it. Okay. I hope this concept is clear then. Moving on to the next question, question number 4. Ability of a chromatography column to separate two analyte peaks from one another is called dash. Option A, distribution, option B, partition, option C, separation, and option D, resolution. Ability of chromatography column to separate two analyte peaks from one another is called. Okay, Shaina answered option D. Rajeshri answered C. Okay. Anyone else wants to answer? Ability of a chromatography column to separate two analyte peaks from one another is called. It is actually called resolution. So Shaina, you are correct. So option D is the correct answer. So what does resolution mean? Basically, if I have a chromatogram, so whenever I am doing a column chromatography, whatever is eluting out of the column is going to a detector, right? And detector, whatever detection method we are taking, so detector will detect the compounds and give the peaks for each compound. Okay, so let's say I have a mixture of 4 compounds A, B, C, D. Okay, so this column is able to separate the A, B, and then we have, let's say, C and D. We have a graph like that, something like that. Just extend this. So here what we can see, each peak represents each component. So we have a peak that is representing component A, then we have a peak that is representing component B, then we have a peak that is representing C and D. Now if you see, the peaks A and B are well resolved. What do you mean by resolved? You can see there is a separate Gaussian peak for A and there is a separate Gaussian peak for B. But if you see, for the component C, what is happening? We have the Gaussian plot like this, and for component D, we have the plot like this. So that means the plot of C and D is overlapping. That means the column is not able to separate C and D as efficiently as it can separate A and B. So this ability to separate these two peaks is basically called resolution. So we will say column is able to resolve between A and B, but column is not able to give good resolution between C and D. Okay. So do you understand the meaning of resolution? We sometimes say the resolution of this photograph is not good. Basically, resolution means the minimum distance between 2 points. Understand like that. So in your daily life, you use the terms like the photo is pixelated, the resolution is not good. What do you mean by that? Pixels, the general photographs that we take, most of them are made of pixels. Pixels are small dots. Now those dots, the smaller the distance between these two dots, the clearer your picture will be. So more number of dots means better resolution. So simply in those terms. So here also the same thing is happening. The better distance these two peaks will have, that means they are well resolved. more overlapping between these two peaks will show a bad resolution. Okay. So same thing applies here. It is also a picture at the end. Chromatogram is at the end a picture of what you are separating. Okay, what your detector has detected is showing you in a form of a picture, that is a graphical representation, right? So that graph is used to see whether the resolution is good or not. So I hope the term resolution is clear first of all, and then what does resolution mean in terms of chromatography? Anyone has any doubt? Okay. So if there is no doubt, I hope this is clear. We'll move on to the next question. Question number 5. In which type of chromatography the stationary phase held in a narrow tube and the mobile phase is forced through it under pressure? Option A, column chromatography, option B, planar chromatography, option C, liquid chromatography, and option D, gas chromatography. This is a very simple question. Which type of chromatography? We will have the stationary phase held in a narrow tube and the mobile phase is forced through it under pressure. Okay, we are getting option D as the answer. Anyone else wants to try? The stationary phase is in a narrow tube. We have been discussing in the previous question. Think up again. Option D is not the correct answer, I'll tell you. Think again who have answered option D, and think who have not answered at all. It is in a narrow tube and I am adding the mobile phase from the top under a pressure, like I am adding. Yeah, HPLC is liquid chromatography, but then what type of chromatography it is? Liquid chromatography or gas chromatography is just based on what mobile phase you are using. If you are using liquid mobile phase, it is liquid chromatography. If you are using the gas as mobile phase, it is gas chromatography. But then both of them have a specific type of chromatography. The answer is, correct answer for this question is option A, column chromatography. What do you mean by a column? I thought everyone would be able to answer this question. So basically column, so till now I was drawing the rough diagrams of column and thinking that you know what a column is. So any narrow hollow tube is can be used as column. So even in HPLC or gas chromatography we use columns. Okay, in a narrow tube we have the stationary phase packed inside. Either you take a pre-packed column or you can pack it yourself. So in a narrow glass tube or a plastic tube, you can fill the stationary phase, pack it tightly, and then you will have the mobile phase entering the column under pressure. Okay. So when the pressure is applied, depending on how much pressure you are applying, it can be FPLC, HPLC, GC, and what the mobile phase is. So if you are using liquid mobile phase, it can be liquid chromatography. If you are using gas mobile phase, it can be gas chromatography. But there's a column chromatography, planar chromatography is totally different than these two terms. So column chromatography is basically a chromatographic technique where the stationary phase is packed in a cylindrical tube and mobile phase is poured over the column. Planar chromatography, on the other hand, you have a thin layer of either paper or a thin layer of silica coated on a medium that can be used as the stationary phase, and the mobile phase will rise up the stationary phase. So in a column chromatography, you will have a cylindrical column. So here the opening will be there, and you can have either, if you are doing manually, then you can have something like this where a tap will be fitted, and if you are doing HPLC or GC, then this system will be closed and you will have it connected to the system, and you can pour the mobile phase in this. The stationary phase will already be packed inside this hollow cylinder tube. In case of planar chromatography, let's say paper chromatography. So we have a piece of paper where you will spot the mixture of analytes. So we have a solution where A and B components are mixed. So I will put a spot, dry it, and then dip this paper in a beaker containing the mobile phase. So I'll dip this paper or TLC, whatever you are using, in a paper containing the mobile phase, where the mobile phase won't be touching the spot that I have made. So mobile phase will be below the spot. Once I dip it, due to capillary action, the mobile phase will rise up. Once the mobile phase reaches the top of the paper, we take out the paper, and then you can observe the separation of the spot. So the spot will separate into two analytes A and B based on its affinity to the stationary phase, how much it. So if A is interacting more with the stationary phase, it will move slower than B. So basically based on the partition coefficient, the separation will happen. Okay, so that is planar chromatography, and this is column chromatography. Liquid and gas chromatography is just a term that is used based on what mobile phases you are using. Okay, I hope this is now clear to everyone.
Okay, ma'am.
Yeah. So moving on to the next question. Question number 6. In a column chromatography, the number of distribution planes is directly proportional to dash and inversely proportional to dash. So option A is difference in retention time and width of the peak. Option B, width of the peak and retention time. Option C, height of distribution plane and height of the column. And option D, height of column and height of distribution plane. So number of distribution plane is directly proportional to what and inversely proportional to what? Anybody wants to answer? Yeah, do you have a question? I see a hand raised. You can unmute and ask. Okay, you wanted to answer option C, height of distribution plane. It is directly proportional to height of distribution plane and inversely proportional to the height of the column. Okay, anybody else wants to answer? We have option A, difference in retention time, width of the peak. Okay. So actually the answer is option D, height of the column. It is directly proportional to height of the column and inversely proportional to the height of distribution plane. So what are the distribution planes of the column chromatography? I'll explain. So in a column chromatography, each thin plane of the column matrix participates in the distribution of molecules. So what do you mean by that? Basically, again I'll draw a rough column. So basically you have this column, right, and it is and it is packed with your stationary phase. Okay, this column is packed with your stationary phase. Now when you are packing the stationary phase, you can imagine like separate this stationary phase in very thin sections. Okay, separate this stationary phase in very thin equal sections. So these sections will be called as plane of the column or column plates. Different names are there. So distribution plane, or sometimes they are called the plates. Okay, so plates of the column or planes of the column. So these thin planes of the column matrix, they are basically responsible for distribution of the molecule. So number of these planes will be equal to the total length of the column. So this will be the length of your column divided by height of each plane. So this is the height of one plane. So height of each plane. So simply if you multiply all this, you like if you add up all this, you will get the total length of the column, right? So basically if you have to tell the number of distribution plane, that would be length of the column divided by the height of each plane. Okay, why this is important, I'll tell you. So if I have a mixture that I want to separate, if I want to separate two analytes from a mixture, the more the number of distribution planes, the better resolution I will get. How? The more number of distribution planes, that means we can actually divide the stationary phase in those many sections. That means if my two analytes now A and B, let's say A have a partition coefficient of 10 and B has a partition coefficient of 5. Now when A is interacting with the stationary phase, it is getting those many sections to interact with the stationary phase. So if the partition coefficient of A is 10, that means 10 parts of A will interact with the stationary phase, whereas only 5 parts of B will interact with the stationary phase. So now A will interact more, B will interact less, but that to get that fine resolution, remember that I told you about resolution. So give that proper resolution, the proper separation of A and B. Like I don't want two overlapping peaks. I want A separated and B separated. Like I don't want any part of B in the fraction where I am collecting A, and I don't want any part of A coming in the fraction where I am collecting B. So the resolution between the two analyte will be good only when we will have more number of distribution planes. So more the number of these distribution planes. So this is a hypothetical concept actually. There are no actual divisions in the column. Column is a continuous thing. Okay, where whenever we have a, like, just imagine a hollow cylindrical tube where you are pouring a slurry of some resin. Okay, so that will be a continuous thing, but it's a hypothetical concept where we imagine that that particular stationary phase is divided into number of planes. So this concept comes from the resolution only. So better the resolution, that means there are more number of distribution planes. So when you keep on using the same column over the time, we will see there is a decrease in resolution. That decrease in resolution is due to decrease in these number of distribution plane. And how can that happen? The height of distribution plane can increase, length of the column can sometimes decrease because of the compression of the bed. So these problems can actually lead to less number of distribution planes and hence less resolution. Okay, is it clear what distribution planes are? This is an important concept.
Okay, ma'am.
Yeah. Okay. So let's move on to the next question. Question number 7. Ion exchange chromatography separates the analytes based on dash of the analyte. Option A, hydrophobicity. Option B, surface area. option C charge, and option D size. Ion exchange chromatography separates the analytes based on dash of the analyte. Okay, we have the answer option C, that is charge. Okay, that is the correct answer. So, by the name itself you can see it is exchanging ions. So, any ionic compound is basically charged. So, that is the simple concept behind it. So, any charged molecule based on its charge, either it will interact more with the stationary phase or the mobile phase, and depending on that we can separate two analytes. Okay. So, if I have a negatively charged stationary phase and I have a mixture of a positive and negatively charged compound, the positively charged compound will bind to that stationary phase, and the negatively charged compound, since it will repel, it will get eluted out of the system. Then we can separate the binding of the second analyte that was positively charged by changing the charge on the molecule. Okay. So, that is the simple concept between ion exchange chromatography. You have different charges, differently charged compounds, and you can separate them based on their charge by using a particular resin that is either positively charged or negatively charged. So, depends on what you want to purify, what you want to extract. Based on that we will choose the resin. Okay. Let us see the next question, question number 8. In which type of chromatography interaction between matrix and analyte is depends on isoelectric point? In which type of chromatography the interaction between matrix and analyte depends on the isoelectric point? Option A, size exclusion chromatography. Option B, ion exchange chromatography. Option C, affinity chromatography, and option D, hydrophobic interaction chromatography. So, which of the chromatography the interaction is based on isoelectric point? Okay. We have one answer option A, one answer is option B. Anyone else? We have more majority on option B, that is the correct answer, option B. So, size exclusion, the separation is based on size, as the name suggests. So, bigger molecules will elute first, smaller molecules will elute later, that is the principle. We will study more about size exclusion chromatography. Ion exchange chromatography, as I told you, the separation is based on charge. Affinity, based on the affinity, something that is more that is having a higher affinity towards the stationary phase, that will bind strongly than the other. And hydrophobic interaction is based on hydrophobicity, that is also apparent from the name. So, here the separation is based on charge. So, what is the role of isoelectric point in charge? So, for especially for proteins, if you know, for amino acids and proteins, the isoelectric point basically is the pH. Isoelectric point is that pH at which the charge is 0. It is a neutral molecule at which charge is equal to 0. So, Pi is that particular pH at which charge is equal to 0. Okay. So, any pH, if I choose any pH below or above Pi, my molecule is going to be charged. Either it is going to be negatively charged or it is going to be positively charged. So, based on that particular charge, I can actually use ion exchange chromatography. So, if it is negatively charged, I will use a positively charged resin, and if it is positively charged, I will use negatively charged resin. So, ion exchange chromatography actually depends on the isoelectric point, because isoelectric point is the factor that ultimately influences the charge on any analyte, and the separation is based on charge. So correct answer is option B. So this is how ion exchange work. So we have two types of we have two types of ions, right? Cation and anion. So positively charged ions and negatively charged ions. So for positively charged ions, we will use the matrix that will be negatively charged, and for negatively charged ions, we will use the matrix that is positively charged. So this is the matrix to which a negative charge is there. So this is a example of cation exchange chromatography, and a cationic molecule will be bound. Once I load the protein which has a positive charge, that means it is having the pH less than its pi. Okay, so if it is having pH less than its pi, that means it is positively charged. If it is positively charged, it will actually have an affinity towards this negatively charged. You know, positive and negative can bind together, and positive will repel positive, negative will repel negative. Okay, so it will kind of compete with this sodium ion that is bound to the matrix right now, and it will replace the sodium ion and get bind with the negatively charged matrix. Then what we will do, we will increase the concentration of sodium ion. Now since more and more sodium ions are entering the system, what will happen, we will increase the salt concentration. Basically we will add more and more NaCl. NaCl in the solution will be in form of sodium ions and chloride ions. More and more sodium ions are there. That means now in the competition, sodium is winning. So sodium can actually break this interaction and compete with the protein. So that's how first we will bind the protein, where we will bind the protein at a low sodium ion concentration. So at that time protein will be higher in amount, so protein will win and protein will bind to the matrix. Then what we will do, we will slowly increase the sodium ions. So the point where the sodium ions are successful competing with this protein, the protein will be eluted out of the column, and we can get purified protein from the mixture of different proteins. Okay, same concept goes for anion exchange chromatography. The only difference is the matrix is having now positively charged. The matrix is having a positive charge. So since the matrix is having positive charge, chloride ions are competing here. So here the negative charge matrix had an affinity towards positively charged sodium ions, and the competition was between the positively charged protein and the sodium ions. In an anion exchange chromatography, we will have the chloride ions competing with the negatively charged proteins. Okay, the matrix will be positively charged, and there will be an exchange of the anionic compound. That's why the name cation exchange or anion exchange. So here the cations are getting exchanged within the matrix, right? First sodium was exchanged with the protein, and then sodium came and replaced the protein and bound with the matrix. Similarly, chloride and the negatively charged protein is competing with each other, so they are getting exchanged in the whole process. So that is why ion exchange chromatography. Okay, and how does pi influences the charge? Basically think of pi as a wall. Okay, so if the pH is on the left side of the wall, that means everything is protonated. Okay, and if the pH is on the right side of wall, everything is deprotonated. Okay, this is a simple way to remember. Now if things are protonated, that means H plus ions are there. Sorry, H plus ions are there. If things are protonated, the protons are loose, that means there will be a negatively charged. So pH less than pi will have a positive charge. So thus you will do cation exchange chromatography. pH more than pi will have a negative charge, and thus you will do anion exchange chromatography. I hope this is clear to everyone. So this is just to make you understand, like whenever you have doubt, like whether the particular pH is giving positive charge or negative charge to the protein. If you know the pi of the protein, you will understand that that particular if the pH is less than that particular pi, that means it has positive charge. If the pH is more than that particular pi, that means everything is deprotonated, so protons are gone, positive charge is gone, that means negative charge. Okay, so remember it like that. Okay, if there are no doubts, we will move to the next question, question number 9th. In a cation exchange chromatography, option A, the column is packed with the positively charged beads. Option B, column is packed with the negatively charged beads. Option C, negatively charged proteins binds to the positively charged column. Option D, both positive and negative charged beads are present on the column. So I just now told you what a cation exchange chromatography is. So I want the answer from your side only. Just showed you the image and explained you how a cation exchange and anion exchange is working. So what kind of bead will be there in cation exchange? Whether it bead means the matrix, the stationary phase. So it will be positive, it will be negative, or what is happening? Tell me. Yeah, option B, the column is packed with negatively charged beads. Yes, you saw the, when I say cation exchange, many people get confused that the name is cation, so that means the matrix will be positive. No, that is not the case. The name cation means the cationic molecule will bind to the column. So column has to be negatively charged and the cations will compete with each other. So positively charged analyte and positively charged sodium ions will compete with each other. Okay, the bed will be negatively charged. Okay, the matrix, the bed, the stationary phase, the beads, whatever you are using, whatever term you are using, that will be negatively charged. So for cation exchange it will be negatively charged, for anion exchange it will be positively charged. So it is always opposite of the name. So remember that. So correct answer is option B. Column is packed with negatively charged beads. The next question, question number 10. DEAE Cephedex column is used for purification of option A, basic proteins, option B, acidic and basic proteins, option C, acidic proteins, and option D, acidic and neutral proteins. So DEAE Cephedex column is used for the purification of. Anyone wants to answer this? No one? Take a guess. Okay, I'll give you a hint. DEAE Cephedex column is a anion exchange column. Okay, DEAE stands for diethyl aminoethyl. So now tell me what type of proteins will bind to the anion exchange column. Okay, we have one answer, option C, acidic proteins. Okay, anyone else wants to answer? Okay, another answer also is option C. So now I have told you it's an anion exchange column, so you should be able to understand what is binding. So option C is the right answer, acidic proteins. So option C is the right answer. So just now I told you anion exchange, that means negatively charged proteins will bind, right? DEAE Cephedex is an anion exchange column, so negative proteins will bind. Generally for any purification process we assume the purification is happening around pH 7. Okay. So, all the acidic proteins will have a low pI. Okay. The pI will be acidic, the pI will be less than 7. For basic proteins, the pI will be more than 7, right? So, for a pI less than 7, I told you, imagine pI is a wall. So, if the pH has crossed the wall, now my pH is around 7, no one will do the purification at a very acidic concentration, because generally things get denatured and it's not very suitable for the system to be handled at a very high or very low pH. Okay. So, let us say somewhere around 7 to 8 or 6 to 8 pH, it is going to cross that pI wall, right, because this pI is acidic. So, I told you, once the pH has crossed this pI wall, that means the charge is negative, and if the charge is negative, we will use anion exchange. Okay. Now, for basic proteins will have a high pI. It will have pI like 10, 11. So, for high pI, the pH 7 will be on the left side of the wall. It hasn't crossed the pI wall. That means at this stage we will use cation exchange, because we will have positive charge on the protein. Okay. So, DEA is FedEx. DEA stands for diethyl aminoethyl. Okay. So, it is a positively charged, it has amino group. So, it has something like NH4 plus kind of a group. So, positive charge is there on the nitrogen, the amino group basically, and that positive charge binds to these negatively charged proteins. Okay. So, you can Google the structure of diethyl aminoethyl, you will understand there is a positive charge on that matrix. Sorry. There is a positive charge on that matrix, and that positive charge binds to a negatively charged protein. Okay. So, acidic proteins are generally negatively charged at a neutral pI, and basic proteins will have a positive charge at a neutral pI. This is clear? Any doubt till now? Whatever we have discussed, anyone has any doubt, then you can ask me. Okay. Moving on to the next question. Question number 11. Which of the following technique is best suited for initial purification of untagged protein from mixture of various plant proteins? Which of the following technique is best suited for the initial purification of the untagged protein from mixture of various plant proteins? Option A, gel filtration chromatography. Option B, ion exchange chromatography and hydrophobic interaction chromatography. Option C, hydrophobic interaction chromatography, and option D, affinity chromatography. So, we have one answer, option B, ion exchange and hydrophobic interaction. Okay. Anyone else wants to answer? So, option B is the correct answer, ion exchange and hydrophobic interaction chromatography. So, basically, if I have a mixture of different various plant proteins and I want to purify a particular protein out of that mixture, what I'll do is, and you see there is mention of untagged. Basically, we add different kinds of tags so that we can do affinity chromatography. So, you will study about affinity chromatography in detail later. So, since it is untagged, affinity chromatography is out of the picture. Now, these three techniques are there. Now, if I have a mixture of different plant proteins, there is a chance that my protein of interest and the other proteins that are present, there can be two or more proteins that have the same size. So, starting with gel filtration chromatography is not a good idea, because if you start with gel filtration chromatography, there will be a large number of proteins, and if you load so many proteins on gel filtration chromatography, you will not be able to, first of all, find out which protein is yours, and second of all, you will not get a good resolution between different proteins. Okay, it's not like different proteins are very different in size and they will elute as single peaks in the whole column. Okay, so you will get very bad resolved peaks. Okay, so that's why gel filtration is not a good option. So we have ion exchange and hydrophobic interaction. Only hydrophobic interaction will also not give you that level of purification. If you combine two or more chromatographic techniques, it's always better to get a better, like it's always a good option to combine two or more chromatographic techniques in order to get good purification. So, if I have a mixture of plant protein, first I will do, let's say, ion exchange. So, what will happen? I will take the whole mixture at particular pH. Let's say I am taking the mixture at pH 7.2. Okay. So, what will happen? Some proteins will have positive charge, some proteins will have negatively charged. Some proteins will be negatively charged, right? So, I am doing, let's say, anion exchange. Okay, I am doing anion exchange chromatography. So, anion exchange, that means the bead is, the matrix is positively charged, right? The stationary phase is positively charged, and the negatively charged proteins will interact. Positively charged proteins will simply elute out. Now, let's say my protein of interest is positively charged. So, what will I do? I will then take the first elution, the starting elution that has not interacted with the column at all, and load it in a cation exchange column. So, cation exchange will have negatively charged matrix, and this positive charged protein, the mixture of different positively charged proteins will be loaded, and based on the solubility and based on the interaction, my protein will elute out at particular time point. So, when my protein of interest is eluting, at that particular time point, there can be some other protein contamination as well. So, to remove those particular proteins, sorry, just give me a second. So, to remove those particular proteins that have eluted with my protein in the cation exchange column 2, so let's say 2 or more proteins had same solubility, so they all got eluted at the same time. Now, to further separate those, I can move on to a technique called HIC. So, then I can move on to technique called HIC, that is hydrophobic interaction chromatography. This is based on the hydrophobicity. So, the column, the matrix will have a hydrophobic group, let's say an alkyl chain or a phenyl chain. So, a hydrophobic matrix will be there. So, the protein's inner core is always hydrophobic. So, the column will interact with the hydrophobic part of the protein. So, how will this hydrophobic part get exposed? It will get exposed at a high salt concentration. Okay. So, it will interact with the hydrophobic part. So, more hydrophobic will interact more, and the less hydrophobic will elute out of the column. So, based on the different hydrophobicity, I can further purify my protein of interest. Okay. So, basically it is always good to combine 2 or more chromatographic techniques to do the purification of a particular component. Okay. Especially in case of proteins, these techniques are very commonly used, and these are very helpful techniques for protein purification. Let's move on to the next question now. Question number 12. Given below are the steps for preparing matrix beads for ion exchange chromatography. First step is removal of very small particles, second is equilibration with counter ions, and third is swelling of medium. What is the correct order of the steps for matrix preparation? So, I want to prepare the matrix. So, I have beads. What should I do? Should I remove the small particles first? Should I equilibrate with counter ions, or should I swell the medium first? So, what should be the first step? What should be the correct order of the steps? So, anybody wants to answer? Option C. So, first equilibration, then swelling, and then removal of very small particle. Okay. Anyone else wants to answer? Come on, it's not very difficult. Think. If you have gone through the lecture videos, this has been taught. What is the order of matrix regeneration? Nobody wants to answer? Okay, then I will give you the answer. So, the correct order is actually swelling of the medium, then removal of small particles, and finally equilibration. So, it will be 3, 1 and 2. That is option D. So, basically whenever we have these matrix beads, so I told you either you can have cation exchange or anion exchange. So, cation exchange will have positively charged beads, cation exchange will have negatively charged beads, and anion exchange will have positively charged beads, right? So, what we will do, we will first swell the medium. So, swelling of the medium means, so these are generally, the beads are generally adhered to cellulose or agarose based mediums. Okay, so I told you, like I gave you an example of anion exchange, that was DEAE, right? So, if you see the structure of DEAE, it is just diethyl aminoethyl, but this diethyl aminoethyl is either added to a cellulose or a dextran. So, any type of sugar or carbohydrate kind of medium. Okay, so these mediums are basically insoluble in water, and that's how we can make a stationary phase out of these mediums. So, this cellulose, dextran or cefidex, these things have to be swollen. So, cellulose powder generally comes, like DEA cellulose comes as a dry powder. Okay, it's a white dried powder. Okay, for cation exchange also we get like CM cellulose, carboxymethyl cellulose. Okay, so this in that basically carboxymethyl is adhered to onto the cellulose, and cellulose, you know, it is insoluble in water. So, what we do, we soak it in water first, let it swell. So, cellulose will absorb water, but it will not get dissolved in it. So, it will be a suspension of the beads in the aqueous medium. Then you can actually treat it with HCl and NaOH to give it proper ionic strength. Okay, so now these are positively charged ions, so they need counter ions to equilibrate themselves. So, for that, first we will do the swelling with HCl and NaOH. Okay, and generally for anion exchange we use HCl first and then NaOH. For cation exchange, we do the opposite. We use NaOH first and then HCl. Then what we do, once the swelling has been done, so let's say I took 1 gram of this powder. So 1 gram can swell up to 2-3 ml of beads based on the swelling capacity. So 1 gram can go up to 5 ml also. So that would be your column volume. Then what you will do, you will resuspend the whole swollen beads in water, in large volumes of water. So once you resuspend it in water, since these particles are heavier, they will settle down first, and whatever small particles, dirt is there in those beads, in that matrix, that will float, and at that time you can decant the water from the top and then again fill it with water. So do it 2-3 times so that all the dirt is removed out of the beads. Then what we will do, then we have to do the equilibration. So we will have to make sure the column is completely equilibrated with the counter ion. So if it is a positively charged column like DAE, this is positively charged column, we need to balance it with the counter ions, that is chloride. If it is a negatively charged column like carboxymethyl cellulose, we need a positively charged ions like sodium ions to equilibrate the column. So all the DAE groups or all the CM groups in that cellulose should be bound to the counter ions in order to get properly equilibrated. After this, if you want to store the beads, then since we are doing the equilibration with acid and base solutions, NaOH and HCl, what we generally do is we just adjust the pH till neutral and then store the beads. Okay. Other than that, the other option, if you want to immediately use the matrix for your protein separation, what you will do, you will then equilibrate the beads with the particular buffer, whatever mobile phase you are using for the separation, for the binding and for the separation. The binding mobile phase will be added on to the column, so that the whole column is now in the environment of the binding buffer. Okay. So first step is swelling of the medium, second step is removal of small particle, and then we will have the equilibration step. Is this clear?
Yes, ma'am.
Okay, very good. So we will move on to the next question. Question number 13. Interaction of proteins with HIC column involves option A, ionic interaction, option B, hydrophobic interaction, option C, covalent interaction, and option D, hydrogen bonding. I told you the answer in the previous question. Option B. Okay, we have a unanimous opinion that it is the option B, hydrophobic interaction. You are correct. So HIC, the full form of HIC is hydrophobic interaction chromatography. So by the name itself you can understand that the interaction between the protein and the column, basically between the protein that has to be separated and the stationary phase, that is the matrix, column matrix, is hydrophobic. The interaction between them is hydrophobic. Okay. The correct answer is option B. I hope there is no doubt in this. Moving on to the next question. Question number 14. Which type of chromatography can be used for purifying heavy metal contaminated water? Okay. So heavy metal contamination in water will be removed by using which type of chromatography? Hydrophobic chromatography, option B, gel filtration chromatography, option C, ion exchange chromatography, option D, size exclusion chromatography. Okay, option C, ion exchange chromatography is the answer I am getting. option C is the correct answer, ion exchange chromatography. So I explained the concept, I have already explained the concept of ion exchange chromatography, so I hope you understand now. Basically, heavy metal contamination is going to be in the form of heavy metal salts, right? So all these salts will be in ionic form. So to remove any kind of ion exchange chromatography is the best idea, because ionic things can bind to the column and you can get pure water out of it, the elution that is coming out of the column, that will be basically water free of heavy metal ions. Okay, so ion exchange is the correct answer. So this is the process of making deionized water, or converting hard water into soft water. So that ion exchange column, generally strong anion exchanges are used, or strong cation exchanges are used for removing these ionic contaminations. Okay. Moving on to the next question, question number 15. In HIC column, the matrix with aromatic ring makes additional dash interaction. Option A, cation pi, option B, pi pi, option C, polar pi, and option D, anion pi. So in an HIC column, the matrix with aromatic ring makes additional dash interaction. We have one answer, option B, pi pi. Anyone else wants to answer? So what kind of interaction can you have with aromatic ring? What do you mean by aromatic ring? Benzene ring is an aromatic ring. Yes, that is correct. So generally, double bonds, between the double bonds we will have a pi pi interaction. So pi pi interaction is the correct answer. So if our matrix has aromatic ring in it, so it will have an additional pi pi interaction along with the hydrophobic interaction. So there are different HIC column matrix can be of different mediums. Like for ion exchange you had DAECM. These are the different examples of ion exchange matrix. Some can have positive charge, some can have negative charge. Similarly, for HIC column also we can have butyl group, we can have octyl group, we can have phenyl group. So in butyl group it means basically 4 carbon chain is attached. So it's a alkyl chain. So carbon carbon like this, 4 carbon chain is attached. Octyl means 8 carbon chains are attached. Phenyl means it has a benzene ring with OH group attached, right? So that is the phenol group. So phenyl group means it has the O negative or OH group attached. So this benzene ring now will have alternate double and single bond, right? So in case of butyl and octyl, the interaction is only hydrophobic. Okay, the interaction is hydrophobic. For phenyl, we will have hydrophobic interaction, but along with that, we will have pi pi interaction. What do we mean by pi pi interaction? The interaction between the double bonds. So double bonds are called pi bonds, right? So single bonds are called sigma bonds and double bonds are called pi bonds, right? So the interaction between two double bonds is called the pi pi interaction. So along with the hydrophobic interaction, it will have an additional interaction, that is the pi pi interaction. That's why the phenyl cephalose columns, basically the columns that have phenyl group as the side chain, are known to be stronger binding columns. Basically, if I am loading a hydrophobic compound, it will bind more strongly to phenyl than to a butyl column, okay, because of this additional interaction. Is that clear?
Yes, ma'am.
Okay, so let's move on to the next question. Question number 16. How is protein eluted out of the HIC column? Option A, by increasing salt concentration. Option B, by increasing protein concentration. Option C, by changing the polarity of the mobile phase. And option D, by changing the pH of the mobile phase. So I told you HIC is based on the hydrophobic interaction. So you know how binding is happening. Now once my protein of interest has bound to the column, how will I take it out of the column? How will I elute it out of the column? So do I elute it by increasing the salt concentration, by increasing the protein concentration, by changing the polarity, or by changing the pH of the mobile phase? So how can we elute it? Okay, we have the answer option A, by increasing the salt concentration. Okay, what do you think happens when we increase the salt concentration? If my interaction is based on hydrophobicity, what will happen by increasing the salt concentration? People who have answered by increasing the salt concentration, I need a reason. Why do you think that is the correct answer? Any one of you? If I get to know the reason, then I can explain better if that is the correct answer or not. Can anyone tell me why they are answering option A? Okay, I will tell you that option A is not the correct answer. It is actually opposite of the correct answer. The actual correct answer is decreasing the salt concentration, or basically by changing the polarity of the mobile phase. Yes, Rajeshri, you have answered it correctly. By changing the polarity of the mobile phase, that is how we elute the proteins out of the HIC column. So what is happening? We have the hydrophobic matrix. So this matrix have hydrophobic groups, and our proteins have hydrophilic and hydrophobic groups, right? So what will happen? The hydrophobic patches of our protein at a high salt concentration will be exposed out. Okay, so at a high salt concentration, the hydrophobic patches are exposed out and they are binding to the column. Okay, when we are adding high salt to the protein, the hydrophobic patches are exposed out, and that is how they can bind to the column. And when we slowly decrease the salt concentration, basically what we are doing, we are changing the polarity. Right now the environment was hydrophobic, so interaction was happening. Now when we decrease the salt concentration, that means the environment is becoming more and more polar. Okay, water content is increasing. What will happen if the water content is increasing? The protein will again bury the hydrophobic patches inside, and the hydrophilic parts will be outside. And once the hydrophilic parts are outside, hydrophilic cannot interact with the hydrophobic, and thus the proteins will elute out of the column. Okay, so the interaction will be at a high salt concentration where the hydrophobic patches are exposed, and the elution will be at a low salt concentration or at a different polarity where the hydrophobic patches are again buried inside the protein. Is that clear? Everybody who answered option A, it should be clear to you. So this is kind of opposite to what we were doing in ion exchange. So what we were doing in ion exchange was the ions are at a low salt concentration, we had less numbers of counter ions. So let's say I am doing anion exchange. So less number of chloride ions will be there, and thus at a low salt concentration my protein can bind. So at a low salt concentration, protein that is negatively charged, negative proteins will bind. Okay. And then when I increase the salt concentration, slowly what will happen? Chloride ions will increase in concentration and thus protein will elute. Okay. So for ion exchange, elution is done by increasing the salt concentration, but there the salt concentration increase is basically affecting the interaction between the charged molecule and the matrix. Okay. Another way of elution in anion exchange is actually changing the pH. By changing the pH, you can actually change the charge on the protein. So if you binding is basically because the protein is negatively charged. If I change the pH, the protein will lose its negative charge and automatically elute out of the column. So that is what was happening in ion exchange chromatography, IEX. This is HIC. So what is happening in HIC? We are having an hydrophobic interaction. So interaction is based on the hydrophobicity. So you have to basically reduce the hydrophobicity, increase the hydrophilicity. So hydrophobic, hydrophilic, this is basically related to polarity. So more polarity, that means more hydrophilic. Less polar, non-polar, that means hydrophobic. Okay. So non-polar solvents will be hydrophobic in nature, they won't get dissolved in water, and polar solvents will be hydrophilic in nature, they will dissolve in water. Similarly, when we are having a low polarity, binding is happening. When we are increasing the polarity, when we are increasing the polarity, that means we are making an hydrophilic environment, then we will have an elution. I hope this is clear now. We'll move on to the next question. Question number 17. Amino acid sequence and its charge at pH 7.0 of the 3 tetrapeptides PQR is shown below. So we have 3 tetrapeptides here, P, Q and R, and the charge at particular pH 7 is given. So for P, the charge is -2, for Q the charge is +1 at pH 7, and for R the charge is +2 at pH 7. If the mixture of the above tetrapeptide is separated on a cation exchange column at pH 7, which tetrapeptide will elute last? Now what I am doing, I am loading the mixture of P, Q and R on a cation exchange column. So think of the charge on the column, think of the charges, look at the charges of each analyte, charge at P, charge at Q and charge at R, and tell me which out of the following P, Q, R will elute last. We have talked about ion exchange chromatography enough, so you should be able to answer this. It's a cation exchange column. What will be eluted last? Option C, R. Okay. R, that is the most positively charged will be eluted last. Yes, that is the correct answer. So basically, as I have explained multiple times now, so P is having -2 charge, Q is having +1 charge, and R is having +2 charge. Now it's a cation exchange column. What do we mean by cation exchange column? The matrix is negative charge. Okay, the matrix that is there will have negative charged ions, negatively charged groups. So that means positive charge will bind. So the most positive charge will bind more strongly, less positive charge will bind less strongly, and negative charge won't bind at all. So what will happen if I load the mixture of P, Q and R? P will elute first, then we will have Q, and then we will have R, because R is interacting most. So this is basically the partition coefficient. R has a higher partition coefficient with the stationary phase than Q, and then we have P. Okay, so most of you got the answer right. So I am assuming that you understood the concept of ion exchange chromatography clearly now. Anybody has any doubt? People who are not answering at all, let me know if you have any doubt or you are understanding everything. If you are understanding everything, that is well and good. If you have any form of doubt regarding ion exchange chromatography or hydrophobic interaction chromatography, you can ask. In the next class, we will start discussing about the next chromatographic techniques. So it's better if you have any doubt, you can clarify it right now. Everything is clear then? We'll move on to the next question. Question number 18th. Which of the following is not a common elution method in ion exchange chromatography? Which of the following is not a common elution method in ion exchange chromatography? Option A, increasing salt concentration. Option B, changing pH. Option C, increasing temperature. And option D, adding organic solvents. So now think of it in a way that ion exchange chromatography, the separation is based on charge. So elution will be based on charge. So which of the following factors is not going to affect the charge? We have one option, option C. We have another answer as option D. We have majority answers as option C. Okay. Technically, both options C and D is the correct answer. Adding organic solvents won't affect the charge. What will it affect? I told you what will happen. If I add organic solvents, what will it affect? What parameter or what kind of property will be affected by addition of organic solvents? Tell me. Solubility is not exactly the answer. Why is the solubility affected? There's a property. Again, why is the protein getting denatured? There's a particular property that is happening. I just told you. What properties do organic solvents have? What is the difference between an organic solvent and a aqueous solution? Polarity is the factor. Basically, organic solvents are less polar than aqueous solutions. So organic solvents basically will affect the polarity. In turn, it will affect the hydrophobicity. So addition of organic solvent is not the right choice for ion exchange chromatography. That is the right choice for HIC. Okay. Rest, increasing salt concentration. Of course, I told you that if you increase the salt concentration, counter ions will increase, and thus the competition will increase and you will get the elution. Changing pH also I told you. If you change the pH, you are basically changing the charge of the molecule and thus elution. Increasing or decreasing temperature won't really affect the charge, so it is also not a common elution technique. And but addition of organic solvent is definitely not the one, because that is a technique that we'll use in some other chromatographic process. Okay, that property is not being exhausted in the ion exchange. Ion exchange is basically based on charge. Hydrophobic interaction, on the other part, is based on the polarity of the molecule or based on the hydrophobicity of the molecule. So in that case, addition of organic solvents can actually affect the elution. As you answered solubility or protein denaturation, solubility is affected because of the polarity. Things are more some proteins are more soluble in aqueous than organic solvents, because organic solvents are not very polar in nature, and proteins generally like a polar environment. Proteins that are most of the proteins are globular, which has a hydrophilic outer part. So if it has a hydrophilic outer part, it will like to be in aqueous environment. So it does not like organic solvents that are non-polar, that are less hydrophilic than the aqueous environment. Okay, and that is why they denature, because they don't like that solvent and they cannot be in that solvent, and basically they try to unfold themselves to expose the hydrophobic part so that they can interact with the organic solvents, because the hydrophilic outer shell cannot interact with the organic solvents that are present. So that is the reason why solubility and protein denaturation, these things are happening, but solubility issues are happening or protein denaturation is happening. But what is the factor that is affecting solubility or protein denaturation is basically the polarity. Okay, so do you understand? I hope this is clear. Okay. Let's move on to the next question. Question number 19. A gradient elution involves a constant elution buffer concentration, a stepwise increase in elution buffer concentration, a continuous increase in elution buffer concentration, or none of the above. A gradient elution involves what? A stepwise increase in elution buffer concentration. Okay, we have one answer, B. Anyone else wants to answer? So actually the answer is continuous increase in the elution buffer concentration. So basically, when I am saying gradient, it means it is gradually increasing. So if I am doing anion exchange chromatography, I am basically increasing the salt concentration. Now to increase that salt concentration, either I can have a stepwise. So actually the question was linear gradient. I am sorry, this is my mistake. The question should have linear gradient elution involves. So linear gradient elution actually involves a continuous increase. So stepwise increase means, if I, let's say, binding has if I have done the binding at a 10 millimolar salt concentration, okay, then what will I do? From 10 millimolar I will directly increase, like the next mobile phase that I am using will be 20 millimolar, then 30 millimolar, then 40 millimolar. So this is stepwise increase. Okay, whereas in a linear gradient, what is happening, we will have from 10 millimolar to 1 molar, we will have a linear increase. Okay, from 10 millimolar to 1 molar, we will have a linear increase in the salt concentration. So we won't have any breaks. So from 10 we will not jump to 20 directly, we will grow linearly from 10, 20, 30, 40 like that. So that is basically a linear gradient elution, that is continuous increase in the elution buffer concentration. Okay. Moving on to the next question. Question number 20. Which of the following can be used at pH 7 to purify a protein with an isoelectric point of isoelectric point of 4.5? Option A, cation exchange column. Option B, anion exchange column. Option C, either anion or cation exchange column. Option D, neither cation or anion exchange column. So what should we use? We want to purify the protein at pH 7. Pi of the protein is 4.5. Remember the Pi wall? What should we use? Should we use anion exchange or cation exchange? Anyone else wants to answer? So if our Pi is 4.5, remember I told you, if this is the Pi, pH on the left side, that means everything is protonated. Protonated, proton means positively charged, positive charge means cation exchange. pH higher than the Pi, everything is deprotonated, protons are gone, that means it has negative charge, and that means our choice should be anion exchange. So here the pH is on this side of the Pi, right? Pi is 4.5, so pH is 7. So what we'll do? We'll do anion exchange chromatography. We'll use anion exchange column. Okay, so option B is the correct answer. Coming to the next question, question number 21. Salting out is a technique used to increase protein solubility, decrease protein solubility, denature proteins, or purify proteins based on size. What do we mean by salting out? Salting out is a technique that is used to increase protein solubility, decrease protein solubility, denature proteins, or purify proteins based on size. Okay, we have one answer, option B. Anyone else? What do we mean by salting out? So option B is the correct answer. Decrease protein solubility. So basically, salting out, we have two terms, salting in and salting out. Okay, salting in and salting out. So what happens in any protein solution, if you keep on adding salt, initially the solubility will increase. That part is called salting in, and then after reaching a certain point, the solubility will decrease. That part, so if you keep on increasing the salt concentration till a certain salt concentration, the solubility will increase. Then solubility will start decreasing. So when the solubility starts decreasing, the protein will start precipitating out of the solution, and that is called the salting out. So you are taking the protein out of the solution by using high concentration of salt. When you are facilitating the protein to be in the solution by using salt, that is salting in. Okay, so there are two techniques. So salting out basically decreases the protein solubility. Okay, so correct answer is option B. Is this clear to everyone?
Yes, ma'am.
Okay, let's move on to the next question. Principle behind salting out is, so I told you salting out basically decreases the protein solubility, but what is the principle? Option A, competition for water molecules between proteins and salt ions. Option B, formation of salt bridges between proteins. Option C, denaturation of proteins by salt ions, and option D, increased hydrophobic interaction between proteins. What is the correct answer? Okay, Shaina has answered option A, competition of water molecules between proteins and salt ions. Anyone else wants to answer? Shaina, you are right. Option A is the correct answer. Competition for water molecules between proteins and salt ions. So this is the graph I was talking about. So basically, as we keep on increasing the salt concentration, you see initially the protein gets solubilized, the solubility increases. After reaching a certain point, the protein now starts precipitating out of the solution. The solubility of protein decreases. Why is that happening? Initially the protein, you know that protein has a hydrophilic outer part and hydrophobic core. Okay, so this is hydrophobic and the outer part will be hydrophilic. Okay, so what is happening when we are adding less concentration of salt? The water molecules are happily present around the protein like a shell. Okay, it's called a hydration shell. Sorry. So this hydration shell is present. Now as we are adding more and more salt concentration, the salt is now competing with the water molecules. So what will happen? The salt will now expose the hydrophobic core of the protein, start exposing the hydrophobic core of the protein. How it is exposing the hydrophobic core? Because it is replacing the water shell that was present. So around this protein, this blue box that you can see, that is the water. Okay, so this water molecules are basically protecting the hydrophobic core from getting out in the aqueous medium. Now when we are keep on increasing, when we are increasing the salt concentration, what is happening? The salt ions are replacing the water. So salt like ammonium sulfate or ammonium acetate, these ions, the ammonium ions, the sulfate ions, these are what they are doing. They are basically replacing the water molecules. These are replacing the water molecules, and in return what is happening? When the water molecule is getting replaced, the hydrophobic part is getting exposed. Okay, and once the salt concentration is very high, all the water molecules have been replaced by the salt, and the protein hydrophobic core is completely exposed, and protein becomes insoluble in the water, because now hydrophobic part is not soluble in the water. So hydrophobic part gets exposed, the protein becomes insoluble, and protein precipitates out of the solution. Okay, so this is a common technique that is used in protein purification, salting out technique. So generally ammonium sulfate is the common salt that is used for doing the salting out process. Why ammonium sulfate and why not sodium chloride? Because ammonium sulfate actually do not denature the protein. Protein is very stable in presence of ammonium sulfate. It just exposes the hydrophobic core, because of which the solubility decreases and the protein precipitates out, and you can actually centrifuge the whole solution and separate the insoluble fraction from the rest of the liquid. Another part where it is important is hydrophobic interaction chromatography. I told you hydrophobic interaction is basically the interaction of this hydrophobic core with the hydrophobic matrix, right? So for that we need to have a salt concentration where the hydrophobic cores are exposed, but still the solubility is high enough. So here at this point, at the top point, we can see the solubility is high, but now enough salt ions are present that can actually expose the hydrophobic core. That means it can interact with hydrophobic interaction column. At this stage, the solubility has decreased, the protein is precipitated out of the solution, so you cannot do chromatography. Okay. At this stage, the salt concentration is not high enough, so the hydrophobic parts are not exposed, so your protein will not bind to the hydrophobic interaction column. But at this stage, it can bind to the hydrophobic interaction column and is yet soluble in the system. Okay. So this was the last question of the session. Anybody has any doubt in any of the concept? Salting in, salting out, hydrophobic interaction chromatography, ion exchange chromatography, partition coefficient or distribution coefficient. Any doubt in any of the concepts discussed today? Can you explain question number 15 again? Yeah, sure. Question number 15. HIC matrices. Okay. So double bonds are generally called pi bonds. So if you know, aromatic rings are basically benzene, any compound containing a benzene ring. Okay. So benzenes have an alternating single and double bond, right? So if I have a matrix where the benzene ring is the side chain, like it is exposed out of the matrix. Okay. So now I add a hydrophobic molecule. Hydrophobic molecule will interact with this ring by hydrophobic interaction. Okay. Hydrophobic interaction will be there because benzene is also hydrophobic and my molecule is also hydrophobic, so hydrophobic interaction will be there. Additionally, since there is a double bond here, this double bond will have, so double bonds are called pi bonds. Okay, double bonds are called pi bonds. So this double bond will have a pi interaction with the molecule. So let's say now my molecule also has a double bond. My molecule has some double bond character. So this double bond will have a pi interaction with the double bond of the aromatic ring. So that is why, because of this, there is an additional interaction. Okay, the word additional is there because already hydrophobic interaction is there. Hydrophobic interaction will be there in the alkyl chain also, where there is no aromatic ring, only carbon chain is present. This is also hydrophobic, this is also hydrophobic. Okay, but this does not have any double bond, so there won't be any pi interaction, only hydrophobic interaction will be there. But there is a pi interaction happening along with the hydrophobic interaction, which makes aromatic ring containing matrix more stronger binding matrix than the alkyl chain matrix. Okay, is this clear to you, Suman? Anyone else has any doubt, or in any other question? Do you have any doubt? Everything is clear? Okay. If we don't have any doubt, then okay. In HIC, binding of protein depends on the hydrophobicity. That's what I explained, right? So the protein will have a hydrophobic, like, see, you know, there are 20 amino acids, general 20 amino acids, right? So some amino acids are hydrophilic, some amino acids are hydrophobic. Now protein will be composed of many amino acids. Some can have the hydrophobic character, some can have the hydrophilic character. The amino acids that have hydrophobic character can actually help the protein bind to the hydrophobic interaction column, where such kind of groups are present. So these kind of groups are present there. So butyl or octyl or phenyl. So these groups are basically hydrophobic in nature, right? So they can bind, they can interact with the hydrophobic parts of the protein. So that is the property that we have used to purify a protein using HIC. Okay, so it depends on the hydrophobicity. More hydrophobic will interact more with the column, less hydrophobic molecule will interact less with the column. So like in anion exchange, more negatively charged will interact more and less negatively charged will interact less. So if I have a mixture of compound, less negatively charged was eluting first and more negatively charged was eluting later. We had a question like that for cation exchange, right? The +2 was eluting last, +1 was eluting. Similarly for hydrophobic interaction chromatography also, more hydrophobic will elute last, less hydrophobic will elute first. Okay. Anything else? Any other doubt? But what will be the salt concentration? So salt concentration, so this salting out concept is actually utilized there. So salt concentration will be kept high in case of binding, and slowly the salt concentration will be decreased. So what will happen, we will have the salt concentration somewhere here. Okay, if we have the salt concentration somewhere here, that is, it's a high salt concentration. Okay, then we will slowly decrease the salt concentration. When we are slowly decreasing the salt concentration, now water molecules can again replace the salt ions. It's a competition between the salt ions and the water molecules, right? So when we are decreasing the salt concentration in case of HIC, the protein will again become more and more hydrophilic, like protein will come into a hydrophilic environment. Salt ions are replaced, there the hydrate shell will again form. Since this hydration shell has formed, what will happen, the protein, the interaction that was happening between the hydrophobic amino acids and the hydrophobic matrix, that interaction will break and the protein will elute out. Okay, so binding will be at a high salt concentration and elution will be at a low salt concentration. Understood? Any other doubt? Yeah, Rajeshri, you are raising your hand. Do you have a question?
No, ma'am. No, ma'am.
Okay, okay. Anyone else has any doubt?
No, ma'am.
Okay. It's good that you guys had some doubts, so I'm hoping that you are understanding the concept. Most of you are interactive in the class also, so I'm hoping that you are understanding the concepts. These are important concepts, you will use it in biotechnology very regularly, so good that you are understanding. Okay, if you don't have any more doubts, I think we can end this session today. Thank you all for joining. I'll close the meeting now.